other side of the way knowledge and science and our own mobilization can be used to change our lives is what we will listen by Bob Langer. As I mentioned before, Bob has become particularly engaged with many Portuguese researchers over the last years. He was very, very much instrumental in the launching of MIT Portugal joint ventures in 2006 and 2007. And since then, he has very much engaged in training and supporting Portuguese researchers and Portuguese entrepreneurs. Bob, as you may know, has become a leading academic scholar, also the American West Medal of Technology, and we have also awarded this year the Portuguese Science Medal to Bob. He has also engaged in many leading entrepreneurial projects, and himself is engaged in several companies. One of them is Modena, one of the leading companies associated with the attempts to develop a vaccine to prevent and to avoid the propagation of COVID-19. Many scholars in Portugal has been interact with Bob Langer over the last years, but he has also been particularly instrumental in supporting patient innovation and the work Helena Canhão and Pedro Oliveira are promoting at the world level, but which was launched through a Portuguese-funded project some 12 years ago. So again, Bob, thank you very much for supporting all the Portuguese researchers over the last years, and in particular Pedro and Helena, and very much for accepting also the Portuguese Science Medal, and we all are listening to you and to your future developments in the years to come. Thank you very much.
Well, thank you very. Can people hear me okay? So anyhow, it's a real pleasure to be able to speak to you. So I thought I'd start and tell you about how, what we've tried to do in terms of innovation. And my background's very different. You know, I'm a chemical engineer. And in 1974, when I graduated, all my friends went into the oil industry. So I thought I should do that too. But as I started interviewing with them, I wasn't that excited about what they were doing. And I was looking for a way that I could use my background in engineering and chemistry to help people. And I ended up taking a job at Boston Children's Hospital and Harvard Medical School with a man named Judah Folkman. And I was the only actually engineer in the entire hospital. And, by the way, it didn't pay nearly as much as the oil companies. But I was very excited about it because he had an idea, which is shown on the next slide. If we could advance the slide. And his idea was that if you could stop blood vessels from growing to tumors, maybe that would be a way to treat cancer and other diseases. And this slide, this goes back to 1971, actually. What happened, his theory, he had a theory that if you had a tumor cell, it started to grow in 3 dimensions. And if you look in the middle lower part of the screen, what you see is it's sort of plateaued. You get about 1 million cells, it turns out, and it wouldn't get any bigger than that because it runs into a nutrition problem. Cells in the center die because they can't get nutrients or get rid of waste. But his theory was that the tumor made something called TAF, tumor angiogenesis factor, that caused the surrounding capillaries, which normally didn't do anything, didn't grow, to now grow directionally right to the tumor. And what that did is it solved the nutrition problem because that could bring in nutrients and so forth. And then as you see in the upper right-hand part of this picture, the tumor would get bigger and bigger. And on the highest part, you see it's very vascularized. And so the tumor is not only big, but it can spread through those blood vessels. That's a process called metastasis, and that could eventually kill. So this turned out to be an incredibly controversial theory. I thought it was great because I didn't know any biology, but most clinicians and biologists thought it was crazy. But anyhow, I was thrilled, and you know, my job was to prove it. And in so doing, could we isolate substances that could stop blood vessels? So how do you think about a problem like that? Well, first we had to find something that could stop blood vessels. And what I began studying was cartilage. Next slide. I'd get bones from a slaughterhouse, and then I found out where all the bones went, and they went to these meatpacking places in South Boston. And I'd get them, I'd bring hundreds or thousands back, and I'd scrape the meat off them, and they'd look like you see here. And then the upper part is cartilage, and I'd slice those off, pieces off, and I'd extract them with certain solvents and then try to purify them, and I'd get different fractions. But that brings up the next question, which is how do you test this? And if you look back at the history of biology and medicine, a lot of times people find factors. And what's really been key is having a way to have what's called a bioassay, a way to study what those factors are doing. And there wasn't one for this field, so we had to invent it. And the next slide shows what we tried to do. It turns out if you put certain tumors in the eye, like a V2 carcinoma, it will cause blood vessels to grow in the cornea. It takes about 9 or 10 weeks. So what we had to do was, and you could actually measure those blood vessels with an ophthalmic microscope. So we were going to use rabbits, and what we wanted to do was be able to take any of the things in cartilage, all of which were big molecules, like peptides and proteins and so forth, and be able to put them in what we call a slow release polymer that could deliver them for this 9-week or more period. And so we asked different, I didn't know anything about polymers, so we asked different people if they could help us who were really expert in the field. But everyone came back with the same answer that's shown in the next slide, that the big molecules can't get through polymers any more than any of us could walk through a brick wall. The literature, by the way, said the same. Next slide. And the only thing that I had maybe going for me is I hadn't read any of this, so I didn't know any better, so I tried anyhow. Nonetheless, I experimented for 2 years. I found over 200 different ways to get this to not work. But eventually, next slide, I was able to make little microspheres or nanospheres. This is a picture of one on the left and one cut in half on the right. And we published in Nature, next slide, the fact that you could use these to release molecules of any size. And in fact, we also in this same paper, this turns out to be the very first time that nucleic acids like RNA or DNA were encapsulated in materials, little nanospheres or microspheres. And as I'll talk about a little later, that ends up becoming key to what Moderna and other companies are doing. So anyhow, we showed that you could do all this. We even showed, next slide, if you wanted to, that you could get constant release. But when I first started presenting this at scientific meetings, it was really ridiculed. I remember in 1976 giving a lecture at a big chemistry meeting, and I talked about this. I was 27 years old. And when I got done with the talk, a whole bunch of scientists came up to me and they said, "We don't believe anything you just said." They said, next slide, that these large molecules can't diffuse through materials. And we were also using organic solvents. They said that would destroy anything you put in. So, and actually things went downhill from there. First, I tried to get grants from the government, but they have these review panels and they all kept saying, well, Dr. Langer doesn't know anything about biology or medicine, so he shouldn't get these grants. And so my first 9 were rejected very badly. Also, I love being a postdoc at Children's Hospital, but a lot of my friends told me that it's probably not a good idea to do that forever. So I started applying for faculty positions after 3 years. But what happened was, I'm a chemical engineer, so I applied to these faculty positions in chemical engineering, but every single one rejected me. They said the same thing as the grants. They said, well, a chemical engineer does oil. It doesn't do anything in biology. So I didn't get any chemical engineering jobs. And in fact, I was fortunate. I did get a job in a nutrition department. And the reason I got that was because the department head there, Nevin Scrimshaw, he was what I'd call like a benevolent dictator. So he liked me, so he offered me a job, but he didn't bother to ask anybody else what they thought. And that probably still would have been okay, except for one thing, which was the year after I joined, he left. So a lot of the faculty decided to give me advice, and they said I should leave too. In fact, the next slide is from Michael Marletta, who was a very good friend of mine. He was in that department at that same time I was in the 70s and early 80s. And he was giving a speech on me a couple of years ago and talking about that. And I'll just read it. He said one evening he went to a dinner at a Chinese restaurant with me and some senior MIT professors. He said, a senior scientist sat quizzing us while smoking a cigar. When the older scientist heard my concepts for drug delivery, he blew a cloud of smoke in my face and said, you better start looking for another job. And Mike said thought he was in a Fellini movie. Well, I didn't have much choice. They didn't want me, but I was there and I hoped that I could stay, and I kept doing my research. And one of the things we began studying was, since nobody believed this could work, was how did it work? So we started taking thin sections through these materials and visualizing them with what's called a cryomicrotome and then various microscopes. And the next slide shows a picture of one of those materials, ethylene vinyl acetate copolymer, as a model system. And it turns out that if something was even 200 molecular weight or greater, it couldn't go from one side to the other. Then how could these molecules get out? Well, it turns out, next slide, that if you put a model molecule in, like say myoglobin, which is red, and you cut a thin section before any release, you get what we call as a phase separation. So the polymer is white and the drug in this case is red. And if you release this for a year in this particular example, what you see is on the next slide. Left behind are where the drug was, are these pores. And these pores are large enough so that molecules of any size can get through. But it turns out that the pores, the way we made these, are very winding and tortuous. So it takes a very long time for the molecules to get through. If any of you have ever been to Boston, my hometown, I sometimes say it's like driving a car through Boston. And over the years, our students have figured out ways to design these so you can get release anywhere from a day to many years. But because we could do this, we could now go back and try to solve the angiogenesis problem. The next slide shows the assay. And what we did, I actually had something like 100 different samples that I wanted to test. And we did like an N of 20 for each of these. And almost every one didn't work. I think of those as the controls. But one did, and the next slide's kind of graphic, but there's no better way to show it. And these are the rabbit eye assays. This is from a paper we wrote in Science in 1976. And if you look on the left, what you see, this is after 9 weeks, is the sheet of blood vessels growing over the polymer to the tumor. And this happens 100% of the time. But, and in fact, if you looked at this eye 3 weeks later, if you did that experiment, it would be three-dimensional, out of the orbit of the eye, it would actually kill. We sacrificed the animals before that. But on the right-hand side, what was striking, and we saw this, if we could go back, we saw this every time, is that when you put this inhibitor in, you get, the vessels are lower, they're sparser, they avoid the polymer, and over 60% of the time, these tumors never grew. So this was the first time that you could actually show that substances could stop blood vessels. And also it gave assays, bioassays, that people could use in the future to study this. Nonetheless, it took another 28 years from this paper before the first blood vessel inhibitor was approved. And that's because companies like Genentech had great scientists, but spent billions of dollars developing these. But what happened starting in 2004 was striking. And that's shown on the next slide, which is that one after the next inhibitor is approved by FDA, starting in 2004. Avastin, the first one, is probably the #2 best-selling biotech drug in history. But beyond that, they're not only used for cancer. Before this, there were no treatments for eye diseases like macular degeneration or diabetic retinopathy using molecules. This was the first time that happened too. And those drugs like Lucentis and Eylea have also become very, very widely used for preventing and in some cases even reversing those diseases. Well, we also felt, Dr. Folkman and I, that beyond angiogenesis, perhaps these drug delivery systems could be useful for other things. So Larry Brown, one of my students, did it for his thesis that showed that you could deliver like a molecule like insulin with a single injection for over 100 days. And that's shown on the next slide. And then one day, Dr. Folkman said to me, Bob, we should file a patent. And that might seem common today, but then in the 70s, Boston Children's Hospital, where I was, never had a patent. So we filed a patent for the very first time in the hospital's history. And 5 years in a row, the patent examiner rejected it. He said it didn't make any sense to him. And then the people at the hospital started telling me, Bob, you should just forget it. He's never going to allow it. But I don't like to forget it. And I started to think, how could we convince the patent examiner to allow this, legally, of course? And science really wasn't working. So as I told you in the beginning, when I first started doing this, everyone told me it was impossible, it couldn't work. And I wondered, did anybody ever write that down? So you can do what's called a science citation search, meaning I could, and I did this in 1982 after 5 years of getting rejected, and I could go back in 1982 and look at who referenced our 1976 Nature paper and what did they say. And I found things that I never realized even existed. And the next slide shows a great example of that. Here's a review article, it turns out, that had been written in 1979 by 5 of the top chemists in the world. And what they wrote, they're describing this field. And I was sort of shocked when I saw it. I'll just read it to you. They said, generally, the agent to be released is a relatively small molecule with a molecular weight no larger than a few hundred. One would not expect that macromolecules, for example, proteins, could be released by such a technique because of their extremely small permeation rates through polymers. However, Folkman and myself have reported some surprising, so surprising is a great word for a patent examiner, some surprising results that clearly demonstrate the opposite. So I showed that to the people at the hospital. They flew down to Washington, showed it to the examiner, and he said, gee, I had no idea. I will allow this patent if Dr. Langer can get written affidavits, written documents from each of these 5 people that they really wrote that. So I was this young professor. I wrote all these well-known people, and they're all actually nice enough to write me back that they really did write it. And then we got this very broad patent. Next slide. And with that patent, I got involved in starting companies. The hospital licensed it to companies. We teach different companies how to do this. And what that's led to, next slide, are all kinds of products, from microspheres and you need the delivery systems, otherwise the drugs are destroyed right away. So there's treatments here for prostate cancer, preventing bleeding, opioid addiction, type 2 diabetes, different eye diseases, pains and medications, schizophrenia, and so forth. Not only could you do this, it turns out that, as I mentioned to you, that that if you could, we could not only make microparticles and so forth, but you could also make nanoparticles. And I thought what I'd do is show you a video of Nova, the TV show. They came and filmed something in our lab, and they did a much better job than anything I can do. So I thought I would show you the video. I want to make sure the sound is on, but if we could show the video of the nanoparticles that we did. Is the sound on?
Encased in a plastic that releases the drug over time. That in turn gets a special wrapping that disguises the package as a water molecule to fool the body's immune system. And last but not least, the address where it should be delivered, a key that will only fit the lock of cancer cells.
A lot of the clinicians I work with tell me it doesn't really blow up the cell like that, but you get the idea. And these are in various, if we could stay there for a second. And these are used in various types of cancer treatments, but maybe even more exciting is that it's been absolutely crucial to RNA therapies. So Alnylam, a company I've been involved with from the beginning, has used nanoparticles to deliver siRNA to the liver, and they actually got approval from regulatory authorities to treat ATTR amyloidosis, which is a rare disease. And as was mentioned in my introduction about me, is we started Moderna, which is a company that develops messenger RNA drugs. They actually have 14 in clinical trials, one of which is the COVID vaccine. And actually, it's not just Moderna that's using nanoparticles to deliver messenger RNA. Every company that's developing a COVID vaccine, Pfizer, BioNTech, all of them are using nanoparticles to do this, because otherwise the RNA would be destroyed. So that's been very exciting, and I have my fingers crossed that it will hopefully help a lot of people. The second thing that I want to go over, since I was working on materials, I was curious, how did materials find their way into medicine? So I was looking into this one day at the hospital, and the next slide shows how it was almost always done. Next slide. And pretty much what was always the driving force for getting materials into medicine was a medical doctor going to their house to find an object that kind of resembled the organ or tissue they wanted to fix. And then they'd use something from their house. So it turns out like the artificial heart is a lady's girdle material because it has a good flex life like a heart. And a breast implant, one is a mattress stuffing because it's squishy. And that was pretty much all that was done in the 20th century. But I thought we could take more of a chemical engineering approach and ask the question, what do we really want in a biomaterial from an engineering, chemistry, and biology standpoint, and then create it from scratch. The next slide shows an example of what we tried to do. I was very interested, and we still do a tremendous amount of work in our lab on biodegradable polymers. But almost all of these degradable polymers degrade like this. They display bulk erosion, and that could lead to bursts of drug coming out, which for an anti-cancer drug or insulin could be fatal. So we said, from an engineering standpoint, what would you really like? And what we said is on the next slide, surface erosion. So we went to the blackboard. I had some very good chemists and toxicologists helping me, and we wrote different structures. And the next slide shows one of them that we thought would be very good. It's what's called a copolymer. And as you change the ratio of those two units, the CPP and the sebacic acid, you can get different erosion or release rates. And the next slide shows that depending on what you put, what the ratio is, you can get almost any release rate you want, from literally a day to weeks or even years. And since we did that, one of my friends, Henry Brem, he's now chief of neurosurgery at Johns Hopkins, came to see me and he said, well, Bob, could we come up with a better way to treat glioblastoma multiforme? And the next slide shows a little bit of, this was the 1980s, of what we were talking about. Normally, the lifespan for these patients, no matter what you do, is less than a year. But the drug they used at that time was shown on the next slide, BCNU. And it's terribly toxic, and you give it, it spreads throughout the whole body. So the idea we came up with was, next slide, could we allow the neurosurgeon like Dr. Brem to operate on the patient, open up the brain, because they're going to do that anyhow, to take the tumor out as much as possible. But before he closes it up, could he line the surgical cavity with little wafers? Now, normally this drug has a 12-minute lifetime, but here with the polymer, it protects the BCNU from degradation. So you get high concentrations in the brain where you want it, and low concentrations where the rest of the body, where it could cause harm. Now, of course, if you're a professor in the US, you have to raise money. And so I would write grants on this, and to try to get money from the National Institutes of Health. And I always did terrible. The next slide shows some of the reviews. So first, when we wrote this, the reviewer said the polymers couldn't be synthesized. But I had a terrific graduate student, Howie Rosen, who synthesized them. And then people said, well, okay, but then the polymers will react with whatever you put in, because you have these anhydride bonds. But I had another great postdoc, Cam Leung, and he showed that didn't happen. Then people said, well, okay, but in 1985, they said the polymers are fragile. They'll break because they're low molecular weight. But I had another couple of postdocs, Edith Mathiowicz and Avi Dome. Edith is a full professor at Brown now, and Avi is dean of pharmacy in Hebrew University. And they made polymers that were high molecular weight and very strong. And then people said, well, okay, but new materials are going to be toxic. But Cato Lorenzen, one of my graduate students who won the National Medal of Technology from President Obama, showed that wasn't the case. And then people said, well, okay, but even so, the drug won't diffuse far enough to kill the tumor. But Mark Saltzman, who was my graduate student and actually headed up Yale University's first biomedical engineering department, showed that they did. Anyhow, this kept going on until 1996, when the FDA approved it. By the way, if people are wondering how we got funding, we did have patents and we got some corporate money. But at any rate, the FDA approved it in 1996. It was the first time in over 20 years the FDA approved a new brain cancer treatment, and the first time ever they approved this idea of local chemotherapy. Just to show you a little bit about how this looks, the next slide shows a picture. I'm going to go fast because I don't want you to have to look too long at these. Next slide. Usually they put 6 or 7 in and close up the brain. The next slide shows you some of the earliest clinical data, and it's not a cure by any means. It basically can prolong life and relieve suffering, but and you certainly see that on the second graph, the 2-year graph. In fact, a couple of years ago, next slide, they did an analysis of all the patients on over 60 trials. And what you see again is that you do get a significant survival. In fact, this has now been this treatment's now been approved for and used the last 24 years. It's been used in over 20 countries. And maybe not only is the fact that it can help in cancer, but also where it's helped even more based on some of our patents is in heart disease. And if somebody has heart disease, one of the main ways of treating heart disease is to put in a stent. Next slide. It's kind of like a Chinese finger puzzle. And but when you put these in, it can cause proliferation, a problem called restenosis. So what people now do is they take a different anti-cancer drug, Taxol, put it on a polymer and coat it on the stent. And these are used by millions of patients every year to keep blood vessels open. Well, the very last thing I want to talk about is, can we make new tissues or organs? In other words, not only can you deliver drugs, but could you deliver cells? And so this was motivated by work I did with Jay Vacanti. And here's a little baby on the next slide that's dying of liver failure. And the only way a child like that could live is somebody else would die. And Jay once asked me, could we come up with a different paradigm? And what we did is shown on the next slide. This is from a paper we wrote in Science. And our idea is that we could take almost any cell type. We actually wrote this before people talked about stem cells, but you could take any cell type. And if you injected at random, not much would happen. But turns out cells are smart. And if you put cells on a scaffold, they could actually reform structures. People had shown this before we did in basic work, like out at Berkeley. And then you have to grow it in vitro, like in a bioreactor, and you could make literally any tissue. And we use different materials. The next slide shows some of the ones that we would use. Either polylactic glycolic acid, which is FDA approved. We also synthesize brand new materials like polylactic acid colysine. And if you look at that amine group at the bottom, you can link in by covalently different amino acid structures, which can help certain cells stick. So we actually had a whole library of different materials. And then you'd make them into fibers like you see on the next slide. Or you could even make any structure by what are called CAD-CAM techniques. Prasad Shastri had done some of this. And let's say you want to make a nose. So the next slide's an example of that. You could use computer-aided design to make a nose. And you see that it's pictures of these. It's like 98% porous. And then you could design it by CAD-CAM, by computer-aided design, and then do something like foaming techniques or three-dimensional printing, and you could make the nose, and then you could take somebody's own cells, like from an ear, and make a tissue. And actually, to that end, some of the people in our lab and the Vacanti brothers, they've actually made tissue. The next slide is a picture of a nude mouse, and where on the bottom part, you can see we've redone this guy's skull. And on the next slide, we've redone this, on the top left, we've redone this guy's cheek. And if you open up the animal and look at the tissue, it's pure white glistening cartilage. You see that on the next slide. And histologically, it's cartilage as well. The only thing is it's good cosmetically, but it's not strong enough if some of you are runners. And so we're still working on that. But what happened is the Army in the United States approached Jay, myself, and Linda Griffith, one of my former postdocs, and they said, we have people coming back from Iraq and Afghanistan, soldiers, without ears and without other body parts. Could you make those? So the next slide is a human ear that Linda made on the top part. And on the bottom right, you see a scanning electron micrograph of polymer fibers that will degrade actually into water and carbon dioxide. But before they do, you put cells on them, and the cells actually grow and they make matrix. And you can actually make an ear. This is shown on the next slide on a rabbit. Next slide. And the reason we do that is we want to show that these are safe for people. So, this has actually now been done on different patients. Here's a little boy who's 12 years old at the time, but he likes to play baseball. But if he were ever hit on the chest with a baseball, he could die. So Jay operated on him. We made a polymer scaffold for him. Jay took his cells and made a chest for him. And he's now doing fine at 6 feet tall as a chest. Other companies have licensed our technology to make new skin, which is actually FDA approved for people who are burned or have skin ulcers. And I'll show that now on the next slide. So this is a little boy who's baby, 2 years old, very badly burned. But the product now is neonatal skin fibroblasts on a scaffold. Next slide. You can put it in the child at the time of injury. Next slide. And so this is time 0, but let's come back 3 weeks later. Next slide. And on the last slide, let's come back 6 months later. Next slide. Yeah. I'm sorry. Yeah, well, we'll stay there. I guess the last slide didn't come out, but basically, he's basically completely healed after 6 months, which is what I was going to show. Now, people are taking this in all different directions. You know, companies and scientists. You know, people are using tissue engineering now to make new spinal cords, to make new blood vessels, to make new corneas. And again, a lot of these are in clinical trials. And it's been taken in lots of other directions too. One of the concerns that people have is like animal testing and human testing. So now what we see is using these kinds of approaches, people having organs and tissues on a chip, which can minimize, I hope, animal and human testing. And people are even doing things that never even occurred to me when we first did this. Like some people, some of my former students even, and others are using this to make meat, you know, from scratch, to make leather and so forth. So this is basically what I want to tell you about some of the work we've done. I'm really deeply honored that you asked me to speak, and very honored about you giving me such a wonderful award. I want to wish you all the best, and thank you so much for having me. Thank you.